Hi, all, and welcome to another session of biology. In today's session, we are going to see a new topic related to our flowering plants. The flowering plants belong to a category called as angiosperms. Flower is the reproductive organ in these plants. So, in today's topic, we will be seeing the anatomy of these flowering plants, that is angiospermic plants. In particular, anatomy involves the tissues, the internal body structure, what is it made up of, how is it arranged. So in angiospermic plants, we'll be seeing mainly meristematic tissue, permanent tissue, and then how the tissue system is arranged within the organs of a plant, that is leaf, stem and root. So if you take a section of root, how will it look? That we'll see under anatomy of root. If you take a section of stem and observe it under microscope, how will it look? That we'll see under anatomy of stem and anatomy of leaf and so on. And at the end, we'll see about a secondary growth which is seen in majority of angiospermic plants. So when I tell about the anatomy, so if we all know human body, so we have the outermost layer, skin. Below skin, we have muscles. Muscles are attached to skeletal system. So if I take the anatomy of a human, we have various structural components. We have skeletal system to which muscle system is attached, muscular system. Then we have skin, epidermis, which belongs to epithelial tissue. Then these tissues are organized to form organ. Organs are organized to form organ system and so on. So this is very easy to understand because we can see and understand the animal body or human body. But when it comes to plants, we think they have three main parts: leaf, stem and root. But if you cut the stem or the leaf, you can actually observe there are several layers or tissues which are present in it. So these layers are in turn made up of the fundamental unit, of course, of our body, that is cell. So just like how animals have an anatomical structure, that is internal body organization, plants also have a structural feature, the anatomical arrangement, how cells form tissues, how tissues are arranged to form particular organ in a plant. So when we talk about tissues, is there a fundamental difference between plant and an animal tissue? So we know, we have already studied in lower classes that plant cell and animal cells have fundamental differences because their mode of nutrition, their way they work is different. If you see plant cell has a cell wall. It has a rigid cell wall which helps in maintaining its shape, mechanical support. It has chloroplast for performing photosynthesis. It has vacuoles which occupies the larger space to store cell sap. On the other hand, if you see the animal tissue, they do not have a cell wall. They have cell membrane as the outermost layer, and they do not have vacuoles. That vacuoles, even if they are present, they are very temporary and minute. Coming to chloroplast, they are completely absent because we are not photosynthetic animals or photosynthetic organisms. So if there is fundamental difference within the major structure, that is cell, so these cells actually form the tissue. So obviously the tissue itself will have difference as well. So if you see plant tissue and animal tissue, depending on the work and the habitat they follow, of course there is difference. Plant tissue are majorly organized for a stationary habit. They do not move much. Whereas animal tissue is organized for more of a motility. Plant tissue, plant body contains majority of its tissues as dead part because it needs mechanical support. If you see trunk, the outer part and the middle part are almost dead. If you see animal, we can't afford to carry dead tissues all around us. So majority of our body consists of living tissues. In plant tissues, we have special cells which divide throughout their life and some tissues which do not divide at all. But in animals, we cannot categorize it because animals stop growing after a certain age, that is after maturation. So you can't categorize animal tissues as dividing and non-dividing as such. So since their fundamental unit cell has certain differences, the tissue and its organization also differs in plant and animals. So when we tell tissue, basically what does it mean is it is a group of cells. So whenever we start the definition of tissue, we'll tell it's a group of cells. So if I put a bunch of cells in a petri dish, will you be able to call it as tissue? Because it's a group of cells, correct? So answer is no, we can't call it a tissue. So we can put hundreds of amoeba in a petri dish, but still we can't call it a tissue, because there are two fundamental points to remember under tissues. That is, these are group of cells, no doubt, which can be similar or dissimilar in structure. So similar, or they can be dissimilar in case of structure wise. Dissimilar in structure. So they can look same together, or they can have different components altogether. But what they should have in similarity is function. Similar in function, that is, they all work together for one goal, and also origin. Their origin has to come from one single source. For example, if I take tissues in humans, there are many tissues. Let me take example of blood. Blood is a fluid connective tissue, but it is made up of many types of cells. We have red blood cells, we have white blood cells, platelets, etc. So structure of cells in this tissue is not similar, but they all follow same function, similar function, that is transporting. They either transport one substance from one place to another. Second function is immunity, etc. Similarly, in plants also we have tissues, and we have categorized the tissues as follows. So tissues are a group of cells. So plant tissue have been divided into two categories. First category is called meristematic tissue. These are capable of division. So they are always dividing. So they divide and give rise to new cells. So they are constantly under cell cycle. Under meristematic tissue also we can divide them into types based on the location as well as origin. So based on location we have three types. One is apical meristem, lateral meristem, intercalary meristematic tissue. Second group is called permanent tissue. Permanent tissue do not have the ability to divide. That is, they have arrested their cell division phase, and they have a particular function to be performed. Permanent tissue can be further divided into two groups. One is simple permanent tissue based on the structure. They are very simple in structure. There are three types under simple. Second is complex. That means they are made up of more than one type of cell. So their structure is very complex. Under simple, three types are parenchyma, collenchyma, and sclerenchyma. Under complex permanent tissue, we have two types, xylem and phloem. So this is the categories of tissues that we can divide within the plant body. So we are going to start with meristematic tissue and then move on to permanent tissue. So first we are going to start with our major tissue, that is the meristematic tissue. As I told, these are capable of dividing. So growth in plants is largely restricted to specialized regions of active cell division called meristems. Meristems means, meristos means divide. So meristematic regions are very confined to one region in plants, whereas in animals, when the baby is born or when the embryo is formed, every cell is capable of dividing. But once the baby is born, only certain cells will divide, and they are not restricted to one region. So you can't find a particular region where you find dividing cells only in animals, but in plants you can find particular regions. For example, if you take a plant body, there are certain regions where you find growth. For example, we have shoot apical meristem, that is the tip of the shoot. where the cell is dividing actively and adding the cells below it. Similarly, root also has meristematic region called root apical meristem. Because root is also growing, tip of the root has dividing cells in it. Then at the middle in the stem we can find lateral meristem, which will help in increasing the width or girth of the stem. Apart from this, you can also find axillary buds like this one, which are in between our main axis and the leaflet. So this axillary bud can give rise to branches. So like this, there are certain regions where you find actively dividing cells in the plant body, and these regions are known as meristems. When we talk about meristem, we also have to see what are the main characteristics. So generally, how do they look? So first thing is they are simple living tissues in growing tips. They have thin cell wall, compactly arranged. Thin cell wall, and cell wall is made up of cellulose. They are compactly arranged because since they are actively dividing every next minute, they can't actually move around a lot, and they have the capacity to divide. And their only function is to divide and add new cells to the plant body. So if you see the meristematic region under microscope, this is how they look. So this is one particular tip of a apical meristem, and you can see it has been stained and focused. So here the pink structures that you see are the nucleus. So what can you observe in the microscope is meristematic tissues are very compactly arranged, very small in size, but the prominent structure that you see is actually nucleus. Cytoplasm is very little, and nucleus occupies the majority of the space in the cell, and nucleus is centrally located. So if you see, there is no space between two cells, that is, they are compactly arranged, there are no intercellular spaces. Space is not seen between two cells. These are the general characteristics. Now if I have to divide meristematic tissues based on certain criteria, there are two main criteria. First criteria is based on origin. So when do they originate in the life cycle of a plant? First type is primary meristem. As the name says, primary, that means it is being formed at the very primitive stage of the plant life cycle. So appears early in the life of a plant and forms primary plant body are called primary meristems. Second type is secondary meristem, of course. Primary meristem is always in active state of division. Secondary meristem appears later than the primary meristem, and it produces secondary tissues like woody areas, and it is called as secondary meristem. Now second basis for dividing is position. So based on position we have three types of meristem. So first type is apical meristem. Apical as in apex, the tip of a plant. So when we talk about tip of a plant, there are two main tips. One is the shoot tip, one is the root tip. So first, they are present at growing tips. Cell division leads to elongation of stem and root and involved in primary growth of the plant. Primary growth refers to increase in height of a plant body. So shoot apical meristem increases the height of the shoot stem. Root apical meristem increases the length of the root, and it can penetrate deep into the soil. Now if you see their structure, so they are localized to apex region, and this is how they look under microscope. So this part is the apical meristem, wherein it has already started to give rise to a leaf, and that region which gives rise to future leaf is called leaf primordia. If you focus on the root tip, so this is the root tip where the meristematic region is found, and there is a cap in the root region, root cap. So this brown part, or the very widely arranged cells are the root caps. So because root meristems are very actively dividing cells, so they have high nutrition value and they are also very sensitive and they do not have very thick cell wall. So when root starts to penetrate deep into the soil, there can be certain mechanical damages that can happen, or certain parasites or pathogens can enter the root because of high nutritive value. So in order to protect this root tip or root apical meristem, root cap is present in the plants. So apical meristem is found in shoot tip, and root tip increases the primary growth of the plant. Now this primary meristem or the primary growth forming tissue also forms axillary bud. What does axillary bud mean is, axil is the angle between upper surface of a leaf stalk and the stem that bears it. So if I take a stem like this and I have a leaf which is something like this, so the upper surface of the leaf stalk and the stem, so that is this angle, this particular region, this is where you will find the axillary bud, and this gives rise to the future branch. So who is responsible for it is again our shoot apical meristem. So if you see shoot apical meristem, so I told this is the meristematic region which is actually continuously dividing and adding cells downward, correct? So the tissue meristematic region is slowly shifting upward, and the already divided cells, the daughter cells, will occupy the below part. Sometimes what happens during this movement of apical meristem, some of the meristematic tissues will be left behind. That is, if you see here, this is the future leaf and this is the meristematic region, correct? So sometimes if they leave behind a certain meristematic tissue, they will localize on the upper surface of the leaf. So if you see carefully, if you leave behind certain tissue like this, it forms a lateral bud, also known as axillary bud, which gives rise to the future branch. Now this is about our first meristem, that is apical meristem. Now coming to the second type of meristem, that is intercalary meristem. As the name says, it is intercalary, in between. In between whom is the question. Now I have taken example of a monocot plant, example sugarcane. So you will find very rigid structures like this here, very solid structures. These are known as the nodes. So this is a node and this is a node, correct? On either side of the node, that means the region between two nodes, is called internode. And on either side of the node you can find the meristem, that is this. So on either side of the nodal region you can find meristematic tissues. So when these meristematic tissues divide, what happens? They add cells, they add more dividing cells to it. So actually it is increasing the growth or height of the plant body in case of monocots or dicot. In dicot, the nodal region is nothing but from where the leaf actually originates. So if the cells in the node region or either side of the node divide, it increases the height or the length of the plant body. So this particular region is called as intercalary. So intercalary meristem is present on either side of the node. So present behind the apex, part of apical meristem left behind during growth period, present at the base of leaf and internode region, leads to increase in the length of the leaf. So this intercalary meristem along with apical meristem increases the primary growth of the plant. Now coming to the third type, that is lateral meristem. Lateral as in sides. So where exactly sides is, they are arranged as cylindrical tissue in the stem. So if you take a section of stem like this section, and observe it under microscope. So you can find a ring-like structure. This ring is the meristematic region for lateral meristem. So lateral meristem is cylindrical-like arrangement which is seen in the stem. So these are found either in between the vascular tissue or within the vascular tissue. That is, they can be found between xylem and phloem or between one vascular bundle and another vascular bundle. So what is their function in general is they are cylindrical meristem, as I told. They are seen in gymnosperms and dicots. Monocots do not have lateral meristem because you can't increase girth of a monocot. They only grow in height and they are seasonal, so they die. Whereas whichever plant is woody and have strong trunk, which shows increase in girth of a stem, will have lateral meristem. Example for lateral meristem is fascicular vascular cambium, interfascicular cambium and cork cambium. That is, in fascicular vascular cambium is present in the vascular bundle between the interfascicular cambium present between xylem and phloem. And they give rise to the vascular tissues and they increase the girth of the stem and root. So they are basically helpful in secondary growth. Primary growth is basically increase in height. Secondary growth is responsible for increasing the girth of the plant body. So responsible for secondary growth. So these are the types of meristematic tissues based on their position. Now let's see some questions related to meristematic tissues. Question is, tissues are options: group of cells which are similar in origin and function, group of organs which are similar in origin and function, cells which are similar in function but not in origin, groups of cells which are not similar in origin or function. So we know tissues are group of cells which have similar function and origin but can have different structure. So correct answer is option A, which cells are similar in origin and function. Second question, intercalary meristem is related to all except present between permanent cells, primary meristem, increasing the girth of axis, regenerates part of grass removed by grazing herbivore. And we know where is intercalary present. It is present on either side of the nodes and it is responsible for primary growth, that is height of the plant. But it is not responsible for increasing the girth of the axis. It is the function of lateral meristem, not apical nor intercalary meristem. Next question, plant tissues are divided into meristematic and permanent tissues on which of the following basis? Options: whether the plant is a dicot or monocot, whether the cells being formed are capable of dividing or not, position, origin. So we know meristematic cells or tissues are capable of dividing, whereas permanent tissues have achieved a permanent shape and function. So they do not divide. So the basis for their classification is option B, whether the cells being formed are capable of dividing or not. So this was about our first set of tissues in plants, that is meristematic tissue. Now we will see the second set of tissues in plants, that is permanent tissue. So what does permanent tissue mean? So we know meristematic tissue is dividing. So one cell divides into two cells gives rise to 4 and 4 into 8 and so on. This is function of meristematic tissue. Where does permanent tissue come in action is one of the daughter cells. So if you take here in the first division, one of the daughter cells is different compared to the parent cell. And if you go further, they have already occupied or achieved some amount of difference from that of a parent plant. They don't look similar, but few of them look similar. That is this one. This is still a dividing cell. because no function, no differentiation. So the process by which a meristematic tissue obtains a specific shape, size and particular function is known as differentiation. And through this process, a meristematic tissue will slowly become a permanent tissue. How? It is a stepwise process, it is not one step process. So first it has to obtain a specific shape which can't be changed later, size and also function. And this stepwise process itself is known as differentiation. So if they ask what is differentiation, it is the process by which a dividing cell obtains particular shape, size and function and obtains a particular function for its lifetime is known as differentiation. In permanent tissue, if you see the characteristics, the cells produced by primary secondary meristem become structurally and functionally specialized and lose the ability to divide. And such cells are termed as permanent cells or mature cells and constitute permanent tissue. So there are two main categories under permanent tissue. One is a simple permanent tissue. Second is complex permanent tissue. So what does simple permanent tissue mean is it is simple in structure. That is, it is made up of only one type of cell. That is, all the cells have same structure. If you see one simple permanent tissue from another, there will be no such differences apart from maybe size, because one is younger, one is older. Second is structurally and functionally they have similar to one another. So we have three types of simple permanent tissue that we are going to study today. One is parenchyma, one is collenchyma and third one is sclerenchyma. So first we are yet to start the simple permanent tissue, but we will see what are the general characteristics of simple permanent tissues. So with simple permanent tissue, one type of cell and they can be either living or dead. First type that we are going to see is parenchyma. Parenchyma, when studied under microscope, it looks something like this. We have two types of sections. One is transverse section, one is longitudinal section. What is the difference is if I take a stem like this and if I cut it horizontally, that becomes a transverse section, and if I cut it vertically like this, it becomes a longitudinal section. So on both the sectional view, we have to observe parenchyma because cells are three-dimensional. They are not 2D, so you can't observe it in one angle and tell the shape of the cell. So if you observe the parenchyma tissue carefully, you can see cell wall. It has a nucleus, cytoplasm, a large central vacuole, and you can also see intercellular space between two cells. They are loosely arranged. So this is, and of course you can see chloroplast in it, some of the parenchymas, not all types. So this is the basic structure. It is a typical plant cell, that is parenchyma. It is found in majority of the parts of the plant body. It is basically like a filling tissue. So wherever you find space between major tissues, you find parenchyma. So main characteristics of parenchyma tissue is, one is they are all living cells. They are generally isodiametric. So they have similar diameter in any angle that you see. So they can be polygonal in nature. Next is, the cell wall is thin and it is made up of cellulose, and they are packed closely. Sometimes they can have intercellular space, sometimes you may not see it. Now if you see parenchyma function, it is the major packaging tissue in the plant body. So it has various functions. First and foremost is storage of food. Since it has a large central vacuole, you can actually store food in it. It can be any form of food, either glucose, carbohydrates, proteins, oils, etc. Second main function is photosynthesis. Of course, they have chloroplast. So in that case, such parenchyma cells are called as chlorenchyma. So a parenchyma having chloroplast which produces or which performs photosynthesis is known as chlorenchyma. Next is secretion. It can also secrete certain secondary compounds like resin, etc. Last but not least function is buoyancy. So in some cases, parenchyma can be arranged very loosely and leaving large intercellular space. So buoyancy is seen when there is large intercellular space. Intercellular space. So this can be seen in only aquatic organisms, aquatic plants like lotus. So in this case, the modified parenchyma is known as aerenchyma. This helps in buoyancy to help the plant float and also acts as a reservoir for gaseous exchange. So these are the functions of parenchyma. Now, this is how a typical parenchyma looks under microscope. Now coming to second type of simple permanent tissue, that is collenchyma. Collenchyma is also very similar to parenchyma. So it looks something like this under microscope. We have transverse and longitudinal section. So first coming to the transverse section. So you can see the large central vacuole. You can see nucleus, cytoplasm is pushed to one end because of the large central vacuole. Cell wall is present and thin throughout except for under the angles where the angles are thick. So these angles are thick compared to the cell wall present in the normal angles. Correct? So if you see the longitudinal section, you will get a better idea. So the cell wall between two cells here is thin, made up of cellulose, but at angles at this position you can see highly thickened cell wall. These thickened cell wall are basically deposition of pectin. And if you see intercellular space, because of this angular thickening, sometimes the intercellular space may be present if it is an young collenchyma cell. And if it is an older one, angular thickenings are very, very huge. So you can't see intercellular space. Coming to the general characteristics of collenchyma, it has cytoplasm, protoplasm, so it is a living cell, but it is a living mechanical tissue. It helps in giving mechanical support to the plant body. Intercellular spaces are absent. Cell wall is thin, but have thickened corners which are made up of combination of cellulose, hemicellulose and pectin, and occurs below epidermis in dicots and absent in monocotyledonous plant. So since it is called living mechanical tissue, because it will facilitate some amount of bending movement for the plant body. But if you see monocot plants, they do not have the ability to bend according to the wind. So because of absence of collenchyma. So since collenchyma is absent, monocotyledonous plants, if you try to bend beyond certain limit, it will actually break. So this particular bending option is given by collenchyma, a mechanical living tissue. Now some of the collenchyma cells can also have chloroplast. In that case, they help in photosynthesis. Now if you see functions, first function, as I told, is major function mechanical support to the growing parts of the plant, such as young stem and petiole of leaf. It helps to resist bending of the stem and tearing of young leaves due to wind. So tearing of leaves can be seen in banana leaf where collenchyma is absent. So a slight breeze or wind can tear up the plant lamella. Secondary function, as I told, if it has chloroplast by chance, it will help in photosynthesis. Now this is how the typical collenchyma looks under microscope. You can actually see the lignin pectin deposition at the angles. Now coming to the third simple permanent tissue, that is sclerenchyma. Sclerenchyma, this is how it looks. You can actually see that only one part is labeled, that too a cell wall. That means the internal structure, the protoplasm, is absent in case of sclerenchyma. And the cell wall, as you can see, is very, very thick. So sclerenchyma characteristic, if you see, they are all dead and without protoplasm. Cells are elongated. Thick cell wall is made up of cellulose, hemicellulose, and main composition is lignin. Lignin is the material which makes it strong, rigid, and impermeable to water. And this characteristic will be used by us for exploiting fibers from the plant body. Now coming to types, there are two main types of sclerenchyma tissues based on their structure, of course. One is sclerenchymatous fibers, which appear to be very thick-walled and tapery end and generally occur in groups. Second one is sclerites, which are also termed as stone cells. These are spherical, overall cylindrical, highly thickened dead cell. That is, even though cell wall composition is same in both fibers and sclerites, sclerites will have comparably thicker cell wall made up of lignin. And these sclerites are the ones which give very crunchy texture to our fruit, very strong structure for a seed coat, etc. So sclerite cells are found in fruit walls of nuts, pulp of fruits like guava, pear, and seed coats of leguminous plants. So this is about our simple permanent tissues. So this is how our sclerenchyma looks under a typical microscope. So apart from these three, if we go to next section, that is our complex permanent tissue. So simple tissues mean they are made up of only one type of cells. Now coming to complex permanent tissue, they are made up of more than one type of cell. That is, even though it's a tissue, it can have 2-3 types of cells, but function should be similar. They differ from one another in their form and structure. However, they work together as a unit. And complex permanent tissues are of two types. One is xylem, second one is phloem. So first we are going to start with xylem. Xylem is the chief water conducting tissue seen in the plant body. And xylem provides mechanical strength to the plant body as well. And if you see xylem function, even though it conducts water, minerals are also transported along with it, and it is unidirectional in nature. So it can only transport water from root to shoot, not in the other way round. Now coming to types of xylem. We have two main types of xylem depending on origin. First is primary xylem. That means it arises early during primary growth and are of two types based on origin. One is protoxylem, primary xylem. Second is metaxylem, which is mature than protoxylem. So this arrangement of protoxylem and metaxylem within the cell or within the tissue is very important. And primary xylem arrangement can be of four types. First one is when we have metaxylem at the center. Metaxylem is in the center and protoxylem is at the periphery. Second type is opposite, where metaxylem occupies the peripheral position and protoxylem occupies the central position. In first case, it is known as exarch type of arrangement. In second type, it is known as endarch type of arrangement. Apart from this, in very few members of angiosperm plant, we can also see the other types, that is mesarch, wherein metaxylem is found on either side of the protoxylem. The last type is centrarch, wherein the central part of the xylem is occupied by protoxylem, The surrounding part is the metaxylem. So if you see the typical stem section or root section, the exarch and endarch can be arranged like this. So this is an example for endarch. This is the example for endarch and exarch having 4 vascular bundles or 3 vascular bundles and so on. Now this is about our primary xylem. If we go to secondary xylem, as the name says, they are formed during the secondary growth, formed by vascular cambial ring, a lateral meristem, and there are 4 main elements of xylem. So we have studied in lower classes the 4 elements: tracheids, vessels, xylem fibers and xylem parenchyma. So here we will be seeing one after the other. First is tracheid. So tracheids have tapery end and they have lignin deposition. So cell wall is made up of lignin. Lignified wall suggests that it is lignin will not allow water to get in. So the tracheid is basically a dead cell, but still it has certain pits that you can see here in the picture like this. So if you observe carefully, the pits are actually where the lignin deposition is very little. So through these pits it can actually transport water. So water can enter and exit through these tips. And when I talk about tracheid, it is one cell. So one tracheid is basically one cell and they are arranged in groups. So one tracheid will have a common wall with another tracheid like this, and another tracheid will come somewhere here. So they form a continuous channel for flowing of the water. And one main thing is, where are they found? They are found in all vascular tissues or all vascular plants. So vascular plants are of 3 categories. So all vascular plants will have tracheids, that is our pteridophytes, the first land plants, our gymnosperms, the seed bearing but naked seed bearing plants, and last our angiosperms. So all these 3 categories of vascular plants will have tracheids. Now coming to second element of xylem, that is vessels. So vessels are broader compared to tracheids. So this is how they look. So as you can see, they have perforated plates. So this is the cell wall of one of the vessel element. So they have arranged 2 vessel element here. So this is one of the vessel element. So these vessel elements have to be arranged one above the other to form a complete vessel. So what we can say is the cell wall of vessels is also made up of lignin, very similar to tracheids. And vessel is not one cell. So vessel is basically made up of many cells, and these many cells are nothing but vessel elements. So vessel element looks something like this. They have oblique cell wall. So obliquely cut or diagonally cut cell wall. And the cell wall is perforated. That means once they were living, and as the vessel becomes more and more differentiated, the protoplasm is lost from the cell and only the cell wall persists. And here again it is a dead tissue, dead cell. And where do you find vessels? Vessels are characteristic feature of only angiosperms. Only angiosperms. Tracheids are seen in all vascular plants, but vessels are characteristic feature of angiosperms. Between vessels and tracheids, vessels are more specialized and more efficient in transporting water. And the 2 main water conducting units in the xylem is tracheids and vessels. Now coming to other name of vessel, it is also known as tracheae. So tracheids are different. Tracheae is another name for vessels. Now coming to the third component or element in xylem, that is xylem fibers. Xylem fibers are sclerenchymatous in nature. Sclerenchymatous suggests it is lignified cell wall, and again xylem fibers are dead. So what is function of xylem fibers is it provides mechanical support to the entire plant body. Mechanical support. And the last but not least, the fourth element of a xylem is xylem parenchyma. It is a type of parenchyma, so obviously it is a living tissue or living cell. So it contains nucleus, cytoplasm. So it is living in nature. And what is its function? It will help in conduction of water, but not in upward direction, but in sideways, lateral conduction or radial conduction of water. So since they help in radial conduction, such xylem parenchyma are known as ray parenchyma. So ray parenchyma are specialized parenchyma found in xylem which helps in radial conduction of water. So this was about our xylem. So if you see the xylem entire tissue together, this is how it looks. So we have xylem parenchyma, vessels which are very broad and wide, and then we have xylem fibers and tracheids. Fibers are one which actually provide mechanical support for the entire plant body. Now coming to the second type of complex permanent tissue, that is phloem. Phloem, as we all know, it is a food conducting tissue, especially the photosynthetic product, which is glucose of course, but when transporting, the glucose is converted to sucrose and getting transported. Once transportation is done, the storage form of photosynthetic product is starch. So phloem carries sucrose from its source to the sink. That is, source is nothing but leaf. It is produced in leaf, and sink can be anything. It can be fruit, flower or root stem. Wherever the energy is required, they will transport the food to that part. Next we have types of phloem. There are two types, primary and secondary. Similar to xylem, primary phloem is the one which is formed first, and it can be protophlloem and metaphloem based on which is formed first. Secondary phloem is formed later and by vascular cambial activity. There is no arrangement as such in phloem, just we saw in xylem. So phloem is very simple. But phloem has particular elements similar to xylem. They also have 4 elements, but they differ in structure and function, of course, because xylem transports water and minerals. Phloem is transporting the photosynthetic product and other food substances. So first element is sieve tube. So sieve tube, as we can see, it's a tubular structure, but again it is like vessels. It's not a single cell. It is made up of certain elements called sieve elements. So here if you see, this is one sieve element. This one particular cell is called sieve tube element, that is one cell, and one cell is arranged above one another to form a continuous tube, that is sieve tube. And between two sieve tube element, that is if I take this is another sieve tube, there is a common cell wall present here. As the sieve tube element becomes more and more mature, the cell wall becomes perforated or porous and form a sieve. So you can see there are pores in the cell wall which forms a sieve-like structure. So which will help the cytoplasm of one sieve element, that is this sieve element cytoplasm, can be in continuous contact with the second sieve element. So cytoplasm is continuous. But what about nucleus? So this particular sieve tube elements, even though are living, they have protoplasm, but lack nucleus. They lack one particular structure, nucleus. So if they lack nucleus, who will guide the cell what to do? We will come to that later. So coming to sieve tube cell wall is made up of cellulose, no lignin deposition, and there will be a companion cell or adjacent cell. As you can see here, we have a companion cell very close, and you can see nucleus in the companion cell, correct? So this companion and sieve tube are always found together. So hence these two together are sometimes called as sister cells. Sister cell. Reason is sieve tubes can't survive without companion cells. Sieve tube has cytoplasm. It is the one which is transporting, but it doesn't have a nucleus. So who guides is the nucleus of the companion cell. The nucleus of companion cell helps in maintaining pressure gradient. Maintains the pressure gradient along the sieve tube. Why is this important is depending on the pressure gradient, the direction or flow of the food will depend. So phloem, unlike xylem, is a bidirectional transporting tube. So leaf produces food, it can get transported to root. Once, if it is required, the root stored material has to be transported back to the shoot. So phloem, my main point is it is bidirectional in nature, and to facilitate it, we have companion cells which maintain the pressure gradient. So based on that, the direction or flow of the materials will be dependent. Now if you see sieve tubes, are they found in all the vascular tissue or all vascular plants? Answer is no. Main thing is sieve tube is present exclusively in our angiospermic plants, that is the flowering plants. Whereas other vascular plants have sieve elements. As I told, tube is the one which contains many elements arranged one above the other. So it's a very sophisticated arrangement. Whereas sieve element is individual, and if it is unorganized, it will never form a sieve tube. So in other lower organisms like pteridophytes and gymnosperm, we can only see the sieve element which is very haphazardly arranged and not very structurized. So pteridophytes and gymnosperms have sieve element, not sieve tube. This is first difference between the vascular plants. Second difference is again with respect to the sister cell, the companion cell. Companion cell is again found only in angiosperms. Whereas gymnosperms have similar cell, whereas pteridophytes do not have any such cell. Gymnosperms have similar cells called as albuminous cells. The albuminous cells help in the maintaining the flow of the elements in the sieve element. So this is about our two main elements in the phloem which act as the main conducting units. Coming to the third element in phloem, that is the phloem fibers. Phloem fibers are similar to sclerenchymatous structure, but they do not provide mechanical support to entire plant body, but in turn provide mechanical support only to phloem. So phloem fibers are also known as bast fibers sometimes, and they are very important economically because of jute fibers, hemp fibers, they all are derived from phloem. Next is our phloem parenchyma. Similar to any parenchyma, has nucleus and cytoplasm. Again helps in radial conduction of food materials. Apart from this, phloem parenchyma can also store certain secondary metabolites and like tannins, etc. And main point under phloem parenchyma is they are absent. They are absent in monocots. Monocotyledonous plants do not have phloem parenchyma. So if you see all the elements of phloem together, this is how they look. We have a sieve plate between two sieve elements, sieve tube wherein cytoplasm can be seen. We have phloem parenchyma. We have companion cells helping, and fibers will be here in case of dicots. In case of monocots, they will be absent. Now coming to the overall structure, if you take section of a stem, so this is how a vascular bundle looks. This is our vascular bundle. That means it contains both xylem and phloem. So if you see, observe the whole vascular bundle closely. So this one is our xylem. You can see perforated cell wall of the vessels. You can see here it is the phloem. Phloem, you can see sieve plate, sieve elements, companion cells. So there is a gap or the perforation between companion cell and sieve element, which facilitates easy transportation of information. And there is one more tissue here between xylem and phloem, that is cambium. So on the right side, you can see the phloem enlarged version. So this was about our complex permanent tissue. Now let's see some question related to these permanent tissues. Question is, major xylem element in wood of a gymnospermic plant is option vessels, tracheids, xylem fibers, xylem parenchyma. So major xylem element in wood of a gymnospermic plant. Gymnosperms we know are below our angiosperms. Angiosperms are more advanced organisms. So gymnosperms in xylem, they do not have vessels, they only have tracheids. So major xylem element in the wood is the tracheid. Next question. Which of the following cells help in maintaining the pressure gradient in the sieve tubes? Option phloem parenchyma, bast fiber, companion cells, wood fiber. So pressure gradient is required to facilitate which direction the food is flowing. So food, even though flows through sieve tube, the main unit which controls it is the nucleus of the companion cell. So answer is companion cell. Coming to next question, component of phloem absent in most of the monocot is option phloem parenchyma, sieve pores, sieve tubes, companion cells. Monocots do not have phloem parenchyma. That is the main difference between monocot and dicot. Companion cells are absent in pteridophytes and gymnosperm but are present in all angiospermic plants. And gymnosperms, even though they don't have companion cells, they have alternate suggestion, that is albuminous cells, to do the function. So if we recall what we saw in today's session, we started with the difference between a plant tissue and an animal tissue. We saw how plant tissue is divided into two groups based on the ability to divide. First we saw meristematic tissue. Meristematic tissues are of three types based on position: apical meristem, lateral meristem and intercalary meristem. Apical meristem and intercalary meristem help in primary growth, that is increase height of the plant. Lateral meristem increases the width of the stem. Lateral meristem is absent in monocotyledonous plant. Coming to permanent tissue, meristematic tissue become permanent tissue by a process called differentiation. Permanent tissue are of two types based on whether they are made up of one type of cell or many types of cells. We have simple permanent tissues. In that we have parenchyma, wherein it's a living tissue, helps in secretion, photosynthesis and also buoyancy and storage of food. Collenchyma have angular thickenings made up of pectin, which is a living mechanical tissue absent in monocotyledonous leaves. Third is sclerenchyma, dead cells which provide mechanical support to the plant. Cell wall is made up of lignin which is impermeable to water. Second group was complex permanent tissue. We have two types, xylem and phloem. Xylem has four elements: tracheids, vessel, xylem fibers and xylem parenchyma. Vessels are characteristic feature of angiosperms. Tracheids are present in all vascular plants. Xylem conducts water in one direction, root to shoot. Three elements are dead: the tracheids, vessels and fibers. Only living element in xylem is xylem parenchyma. Coming to phloem, which transports food, it is a bidirectional flowing tissue. It has four elements, that is sieve tube, companion cells, phloem fibers, phloem parenchyma. Phloem parenchyma is absent in monocots. Phloem fibers, also known as bast cell, have economical importance. Sieve tubes and companion cells are called sister cells because companion cells will help in functioning of sieve tubes. Sieve tubes are living but lack nucleus. So in phloem, three are living, only dead is phloem fibers. So sieve tubes are characteristic feature of angiosperm, whereas lower vascular plants, that is pteridophytes and gymnosperm, only have sieve elements which are not very structurally organized. This is what we understood in today's session. In our next session, we will be seeing the tissue system, how these tissues are arranged within the plant body. Until then, thank you.